In this section, we're going to discuss passive transport. First, let's have a quick discussion on solutions. Solutions are made of solutes and solvents. They are homogeneous mixtures of two or more pure substances. The solute is dissolved in the solvent. Solvents are usually liquids, but can also be gases or solids. Commonly, there is more solvent than solute. In biological systems, solutions are found on both sides of biological membranes. The most common solvent is water. Solutes include gases, ions, and molecules belonging to the four classes of biomolecules. Membrane selective permeability. The important characteristic of membranes is they exhibit what's known as selective permeability, where membranes allow some substances to pass freely while they control the movement of some other substances and prevent the movement of others. They are selective about the molecules that permeate the cell. Membrane transport refers to the selective movement of solutes or water as a solvent across a cell membrane. There are many types of proteins and mechanisms that control membrane transport and maintain selective permeability. Membrane transport is one mechanism that cells use to maintain homeostasis and do cellular work. Membrane transport. The phospholipid bilayer allows the diffusion of gases and lipids. Polar molecules, any charged compound, and ions require transport by specific membrane proteins. There are two types of membrane transport involving the movement of molecules and ions across a membrane. Passive transport, which requires no input of free energy, and active transport, which requires an input of free energy. Passive transport is what we will discuss in this section now. Active transport will be covered later in the presentation. Passive transport is the movement of molecules and ions across a cell membrane without an input of free energy. It may occur when the concentration of a substance is greater on one side of the membrane than the other, and substances move from an area of high concentration to an area of low concentration. This is known as moving with the concentration gradient and can include several kinds of transport. Simple diffusion, facilitated diffusion, which includes the use of channel proteins and carrier proteins, and osmosis and aquaporins. Diffusion is the random movement of particles in gas or liquid from an area of high concentration to low concentration. Particles move randomly in all directions, but if there is an uneven distribution of particles in one solution, then over time, the particles naturally spread out and become evenly distributed. Watch the blue dye diffuse in lukewarm water for 1 minute. Eventually, the dye will be evenly distributed throughout the solution. Here's another example for diffusion. If a strong perfume is sprayed in the corner of a classroom, it will have a strong smell in that corner at first, and it will immediately begin to spread out. Over time, the perfume molecules will diffuse to the rest of the classroom, and more students will be able to smell it. The random movement of the perfume molecules in all directions results in them spreading out over time until they are evenly distributed in space. No free energy is required for this to occur. The distribution of molecules follows the second law of thermodynamics related to entropy. So again, if I have a large concentration of molecules in one part of an area, they will move randomly in all directions until they are evenly distributed. Concentration gradient. Particles move randomly, becoming evenly distributed in their container, with the net direction of diffusion following the concentration gradient. The random movement includes particles moving opposite the gradient, but the net movement is from an area of high concentration to an area of low concentration. Membranes often act as barriers between areas of high concentrations and low concentrations of solute. So here's a membrane. You can see in the extracellular space outside of the cell, we have a very high concentration of this solute particle. If that solute particle, if that molecule is allowed to cross the membrane, remember, not all kinds of molecules can, but if it's allowed to cross the membrane, it will diffuse. And so we see that diffusion happening until the concentration is at equilibrium or equal on both sides. Here's one way that this happens regularly. Oxygen and carbon dioxide molecules are small and nonpolar, so they may cross the lipid bilayer membrane by diffusion. The green dots represent O2 molecules. No energy is required for them to move. They will move in all directions, and their net movement will be from outside to the inside of the membrane, as the O2 concentration, as you can see, is greater on the outside than the inside. The net diffusion of O2 molecules will follow the concentration gradient, moving from areas of high concentration outside of the cell to low concentration inside of the cell. The net diffusion of particles in a solution will be with the concentration gradient. So particles will diffuse with the concentration gradient until dynamic equilibrium is reached. When dynamic equilibrium is reached, particles continue to diffuse in all directions, but the average density of particles in any area remains relatively constant over time. So particles will move from an area of high concentration to low concentration until the particles are evenly distributed throughout. And now the particles don't stop moving here, right? Like one of these particles may move this way, but at the same time, a second particle is moving in the opposite direction. So the average density of the particles in any area remains relatively constant over time. And in a membrane, you can think of like equal number of particles coming into the cell through the cell membrane as particles going out of the cell through the cell membrane. Rate of diffusion. The steeper the concentration gradient, the greater the rate of redistribution. So you can see that when we have this large density of particles on one side of the container here, we have a really high rate of redistribution. And as the particles begin to spread out, that rate decreases with time until we reach dynamic equilibrium, where then the rate of redistribution is none. The speed of random movement increases with temperature, increasing the rate of diffusion. So you can see each of the lines on the graph represents the rate of diffusion of a given carbohydrate in an aqueous solution at different temperatures. And in every case, you can see that as the temperature increases, so does the rate of diffusion. The Fry and Eddington experiment that we discussed in the previous section showed that diffusion of phospholipids in the membrane was also temperature dependent as they repeated their experiment at various temperatures. The rates of diffusion of different solutes are independent of each other. Each type of molecule moves along its own concentration gradient. Some may diffuse into the cell, others may diffuse out. Diffusion is a spontaneous process, and each type of molecule diffuses at its own rate. So let's look at the example above. We have two different gases in two different bulbs, and currently they're separated from each other because the stopcock is closed. If we open the stopcock, diffusion will begin because we have an area of high concentration of O2 here, low concentration here, an area of high H2 concentration here, and low here. So the H2 will diffuse to the right, the O2 will diffuse to the left. And so that's happening. But if you look, the H2 appears to be diffusing to the right much faster than the O2 is diffusing to the left. And that will happen until sometime after the stopcock is open, when dynamic equilibrium is reached. So in that example, the two gases diffuse in opposite directions and at different rates, independently of one another. Facilitated diffusion. Some molecules, like oxygen and carbon dioxide, readily diffuse through all cell membranes because they are small and nonpolar. They can squeeze between the phospholipids. However, larger molecules, polar molecules, and ions cannot squeeze between the phospholipids, and or they're repelled by the hydrophobic membrane interior. As in passive diffusion, particles undergoing facilitated diffusion move with the concentration gradient without an input of energy. Unlike passive diffusion, particles undergoing facilitated diffusion cross the membrane with the help of an integral transmembrane protein. Types of facilitated diffusion proteins. Channel proteins provide hydrophilic corridors that allow a specific ion or small molecule to cross the membrane. So here's a channel protein right here. It crosses the entire membrane and provides a channel for maybe an ion, let's say, to cross the membrane from the outside of the cell inside. Carrier proteins bind to one or a few specific solutes and change shape, helping the movement across the membrane. So look at what happens here on this side. The carrier protein is open for a certain shape, and then it changes its shape to allow that molecule, whatever it is, inside of the cell. Let's look a little closer at carrier proteins. Sugars, amino acids, and nucleic acid building blocks are all relatively large and polar, and they require carrier proteins to cross the membrane. Each type of molecule has a specific carrier protein. When the diffusing molecule binds to the outer part of the carrier protein, which you see here, the carrier protein changes shape and transfers the molecule across the membrane, which you will see here. We will return to carriers later. Some carriers move molecules against their concentration gradient, so they are active transporters rather than passive transporters. Gated diffusion proteins. Gated ion channels, either ligand-gated or voltage-gated ion channels. Although they are small, ions are charged and thus need a channel to cross the membrane. Ion channels are what we call gated, which means that they are usually closed, but a chemical change on one side of the membrane causes the channel to change shape and allow a specific ion to pass through. When a molecule binds to the channel, it opens to allow the second ion to pass through. So when this ion 3, so right now the channel is closed, when this ion binds to the channel, you see those little triangular ions there? That causes it to open and allows the circular ions to pass through. Molecule 3 is what is called a ligand. So this is a ligand-gated channel. It is a molecule that binds to a specific receptor on this channel in order to change its shape and allow it to open. Facilitated diffusion proteins. Channel proteins, such as aquaporins, provide corridors specific for water molecules. Other molecules cannot travel through an aquaporin. The exterior of the channel is hydrophobic, which anchors it in the fatty acid bilayer. The interior of the channel is hydrophilic, creating a passage for a water molecule. The alpha helices of an aquaporin are what create the hydrophilic water channel.